Fabien LEENDERTZ, qui nous vient de l'Institut Robert Koch à Berlin. Donc il est le responsable d'une unité qui travaille sur les zoonoses émergentes. Il est vétérinaire, il est virologiste et il est primatologue. Il a commencé sa carrière en Afrique, enfin sa carrière sur le terrain en Afrique, auprès en particulier des chimpanzés, en Côte d'Ivoire et dans différents pays d'Afrique de l'Ouest et d'Afrique centrale. Il a beaucoup travaillé, en particulier en Côte d'Ivoire. Donc je vais le laisser faire sa présentation. Et bien sûr, il va faire sa présentation en anglais. Il parle très bien français, donc vous pourrez lui poser des questions aussi bien en français qu'en anglais à l'issue de sa conférence. Fabien.
Merci beaucoup. Thank you very much for the invitation. Thank you, Bernard. Thank you, Professor Maul. Thank you. It's a big honor for me when my president heard that I have to give a talk here in Marseille. He said, that's a big honor, you should go there. I was invited to a reception at his house yesterday evening, so I couldn't go there. He said, it's more important that you go to Professor Maul's unit and show him what you're doing here. So I will try to give you a broad overview of what we are doing in the respect of emerging diseases and wild great apes. I won't have the time to go too much into detail in the specific subjects we are working on. As you will see, we are really broad. So I hope if there are questions which are more specific, that there will be time later to discuss those, since I will only scratch at the surface of what we're really doing. So I want to give you a short overview about the concept and questions we have. I will spend some time on material and methods because even though that's not real science, I realize that these informations are always interesting for people to also see if there are points for collaboration and future projects perhaps together. Then I will talk about the great apes in their functional sentinels and sources for infectious diseases. Then I have a section about me. And obviously I will acknowledge some people because I'm not doing the work alone. So our questions are focused around transmission events from animal sources to humans. This is sort of in the core of what we're supposed to study, and this is why the Robert Koch Institute is also paying the salary. However, to understand what is happening here at that interface and why certain diseases are suddenly transmitted to the new host, let's say in this case humans, we need to understand the disease ecology. So circulation of certain microorganisms in the natural community, let's say, of mammals. Since transmission events are not happening always right now, they can happen, but in theory I will say they're rather rare events. We have to take a more evolutionary point of view and look also when in history diseases or microorganisms have been transmitted amongst species. And we can do this through phylogenetic analysis using the range of species we're looking and the species distribution along the geographic range. And we are moving now more and more in the direction of looking at microorganism communities because it's obvious that just looking at individual pathogens will often not explain the whole picture, and also technically these things became more and more possible in the last years. Basically, the concept behind is the One Health concept, where we look at animal disease, human disease, and the environment. We have two approaches in the field. We have a large-scale sample and data collection, which we're doing in collaboration with local partners and international organizations. And this is sort of more a snapshot. These are missions of a year maximum, or a few months in some cases. And these are the countries where we are active, where we have samples and information from. In addition, and I think this is a very important approach, we have a rather small scale but long-term approach, which we are mainly following up in Thai National Park here in Ivory Coast at the border to Liberia. And you will hear more about this area. I'm working in this forest since 15 years now and have a nice data and sample collection in that respect. What kind of samples are we collecting? We're collecting invasive samples from wildlife. So since 15 years, we do full necropsies on every animal we find dead in Thai National Park in the forest. But we're also doing that, obviously, when we do the broad scale in other areas. If there's a dead animal, people are trained to do safe necropsies. In some cases, we collect blood samples, so invasive samples from animals we can't anesthetize, no matter if it's monkeys or duikers, or we are catching also bats and rodents along the gradient from the rainforest into the villages. And now we even made the step into the bigger cities, so we just finished a mission in Conakry looking at rats and dogs and things which I thought I would never work on. However, the most important source of biological samples are non-invasive samples, which we can get from wild-bred apes without disturbing the natural behavior. So we're working with primatologists, we're working on the behavior of the chimpanzees, gorilla, and bonobo. And so we can get those samples from known individuals. We can also get urine sample from the wild-bred apes. We're collecting bones. And another new, I will talk briefly about it, non-invasive approach is to use blowfly as biological material samplers in the rainforests. Since we also have to see if certain microorganisms made the step into the human population, we have to work in the villages as well. So we have bushmeat studies, especially in DRC and Ivory Coast. We are sampling the human population and then the domestic animals around. So it's quite a holistic approach. So over the last years, we have accumulated a nice set of samples together with our Ivorian partners, which we can now use for different subjects. So this was the part about field work, where we have sample and data collection, and where we can, we also get background data on the animals we are sampling, be it ecologists we're working with, or primatologists, where we have the nice data set about behavior. Then we're moving to the laboratory. We're doing some serology. A lot of what we have been doing, and we're still doing in the last years, we are doing it with a PCR-based approach, but we're doing more and more also capture methods and next-generation sequencing, so the broad approach depending on the questions. And I will not go into detail here of what we're really doing, because I realize that in the audience there are people who know exactly how all this works and who are probably even more familiar with those methods than I am. After that, there is the in silico laboratory with the statistics, phylogenetics, and the ever-growing field of bioinformatics. And me being a very normal veterinarian, I really have to struggle with that part, but I have good people in my team who help us with that, and we're doing that also in collaboration with others. Okay, so I want to move to the great apes. So if we think about microorganisms in the wild bred apes, I know it's a little bit simplistic, but this is how I have divided them up now. There are those causing acute or lethal disease, some chronic or non-severe diseases, and those not linked to the disease yet. So I want to start with the top killer for the great apes, which is Ebola virus. And as you all know, the Ebola virus and Marburg viruses. We will now focus here on Ebola viruses. And if you look at what species have been infected today with Ebola, it's the primates, humans, obviously, chimpanzees, gorilla, and some smaller monkey species. With individual cases, no larger outbreaks. Then we have ruminants, we have duikers, forest duikers who can die from it, and bats. So those have been shown to be affected. And I know you know all this anyway, but just to remind, we have now to think about what is reservoir, so a natural host at the origin of an epidemic. What is the host? So someone sustaining the circulation of an infectious agent, or an intermediate host, someone who can be infected, perhaps even die from it, and then be on the origin of an epidemic. So for humans, we know that we are obviously victims, we are perhaps even intermediate hosts, and it had an important impact on the human population in the last years and over the last century. For great apes, the data are not as good as for humans, obviously, but at least from two areas, there are data I would trust much more. which are here lossy, sanctuary in the Decayed Forest, where primatologists knew exactly how many gorillas they had in the area, and then they could observe the decline, and there were samples tested in search and really positive for Ebola. So Ebola had an important impact on these grade A populations. The question why it spreads so nicely, especially in the gorilla, less efficient in the chimpanzees as far as we know, is due to their social structure. As you probably know, the lowland gorilla, they have one silverback male who keeps all his females around, and if this guy is dead, the females will leave that area, the group, within the next hour and go to the neighboring group. So if they were infected, they bring the virus to the neighbors. And then the one who's most interested in the new arrival female is obviously the male, so he gets infected first, and the likelihood that he dies early in that community again is high. So that's creating probably some kind of wave-like spread. In addition, we have video footage that the great apes are really curious when they find a dead animal, and I've seen that myself. They touch them, they sniff them, and obviously that's also a good way to transmit the virus in between the groups. For the chimpanzees, it's less easy because the communities are more stable. There are many males, so if one dies, that doesn't mean that the group will fall apart. So there are these epidemics, especially among the gorilla, and Eric Leroy and his team have done very nice studies, involvement studies on outbreaks in humans and animals, and they can show that actually very often you have an animal outbreak at the same time or just before you have a human outbreak. And this is due to the fact that when people go in the forest, they will bring home any animal they find fresh enough, no matter if they have killed it or they found it dead. And if it's found dead, it bears a bullet. And this is obviously a risk. And now, the bigger the epidemic is among the wildlife, especially the great apes, the higher the likelihood that a hunter goes in the forest, finds a dead gorilla, and gets contaminated. This is why there's a direct link between outbreaks in wildlife, especially great apes, and many of the human outbreaks. So here, the chimpanzees, the gorillas can serve as sentinels for disease outbreaks actually in the humans. So what about bats? Bats have been shown to be PCR positive in these three species here, Epomox panthetti, Mytinax monstrosus, and Myrictolus torquata. This was a nice study done also by Eric Leroy and his team, but they showed only small fragments of the RNA of the bats. They didn't make it to amplify larger fragments or even cultivate any virus. But still, there was something in those bats, and I'm sure these results are solid. Furthermore, we have serological evidence for Ebola virus exposure of bats. We know that some bat species survive experimental infections and can shed the virus. And we know from the little brother of Ebola, from Marburg virus, that bats are permanently infected in certain cases in Uganda with Ebola virus. Like 4% of them carry the virus. And that they are also at the source, at the origin of several human cases or epidemics. Interestingly here, in some of these cases you find fruit bats and insectivorous bats infected with Marburg virus. This is something which people like to somehow not read in their publication where it's clearly stated black and white, but so it's known that those insectivorous bats can be infected with filoviruses in the body. We did a little overview on which species have actually been tested, or which genera of bats have been tested by PCR. You can see here the species richness of these genera, and then here you can see how many have been actually tested. And just looking at this picture, you will see that there's a huge sampling bias, obviously towards the species where PCR results were positive, but to our opinion, the sampling was really not systematic enough, and I think further teams should look at systematic sampling of different bats in general to look for filoviruses. And it's even worse if you look at the picture in the serology where you see some fruit bats tested. The circles are bigger the more are tested, and then we have added a 95% confidence interval. So there are some signals here in the fruit bats. If you look at the number of insectivorous bats and the confidence interval, you will see that nothing is really significant. For Marburg, the picture is a bit more clear, because there we know also what to target, but I'm sure that we will also find other species infected with Marburg virus if we only look for it. Just a few slides about our question when we heard about the outbreak in Guinea, the Ebola outbreak in Guinea. Since we are in Ivory Coast and we have good partners working in Guinea, we thought perhaps we should try to find out where the source of this devastating outbreak was. And we had a very simple question in mind. We wanted first to know if there was an outbreak in an amplifying host. So in the chimpanzee population in Guinea, has the population dropped, and are there still perhaps mortalities going on in the wild chimp populations? Or as an alternative hypothesis, where the perhaps transmissions directly from the suspect as well. So with Sentinels, we put together a team of 70 people from different disciplines to go to Guinea. Some were already in Guinea to look at these aspects. And to sensibilize the whole team, make sure the safety measures are in place, and so on. We did not know what to expect, so we brought all the possible sampling equipment we could think about, and started with the biomonitoring, so to look at the density of chimpanzees in those forests which exist in the forest Guinea area, and in areas where we have good data on how many chimpanzees and other wildlife exist in these areas in the years before from our partners, a conservation organization working there. So we could simply repeat this monitoring and see if there was a drop in animal densities, and what we found is that indeed the populations were rather stable, and that at least there was no huge epidemic in the wildlife. While we were doing this monitoring, the authorities also informed us where actually the index case has lived and who it was. And until now, all the data still point toward that index case, because I know that index cases are just a suspicion. But it looks like that's quite likely really the case. So we went with part of the team to Milyandu, this village where the outbreak started. And Milyandu, if you look here at the map, it's up here. And that's a 4 or 5 hours drive, depending on how the road is at the moment, from the areas where we actually still find chimpanzees and other larger wildlife. Around Milyandu, there is nothing. If you look here, this is a typical landscape around. It's fields, it's a cacao plantation to the maximum. And we talked to the one hunter living in Milyandu, and he says he's only hunting rats, and perhaps once a month he will find a little antelope or something. So a large wildlife outbreak in the area is not possible, because there is not a large wildlife left. So the classic scenario is a hunter who went into the forest to get in contact with a dead animal is rather unlikely. But our anthropologist who was in the team, she discussed with many people and found out that also actually the children are big hunters, and they go hunting with their sticks under the roofs and will find bats there, kill them, and roast them over the fire or play with them. And this is nothing special for Milyandu, for this village. This is something we find broadly in West and Central Africa. And I mean, I have 3 kids at home. When they find something in the garden, they will pick it up and play with it, no matter if it's a worm or a mouse or something like that. It's totally normal kids' behavior. So there is obviously exposure possible. And our question was that if there is anything special about Milyandu, or is it just the normal play behavior and a really, really unlucky event? And so the only thing, and I know that's really shaking a bit vague, but still something to mention, is that in Milyandu there was a tree just next to the path leading here to the water place where the women go for washing, so the kids will always crawl along this path and then play in this nice big hollow tree, which is a nice place to play. I would play there if I was a kid. And that tree, according to the villagers, was full of bats. So there was a huge bat colony living in that tree, but unfortunately the tree was burned a few days before we arrived with the team, so we had no chance to catch those bats. So I would have turned into a famous scientist probably if I would have found finally the source of an outbreak somewhere, but it was burned. So all we could do in the end was to ask people about what kind of bats lived in the tree and try to get some descriptions. And these descriptions were already quite interesting because they said actually it's not bats, it's mice which are flying because they have a long tail and they're also crawling a lot. So that was already very suspicious for us. But then we collected soil samples around and in that tree and control trees obviously and so on around, and did a PCR for mammal DNA and then deep sequencing of these PCR products. And our results showed quite clearly with two different approaches that actually in the tree there was a Mops condylurus, so a molluseed bat, tree-tailed bat, living in that tree. And this is like a control tree with another bat species. And this is also a species which fits to this description of the villagers. I know that that's not proof at all that these species were really at the origin, but it's a hint in one direction. And so we looked a bit more at the literature to find out if these insectivores could be linked to filoviruses. So we have the Smart finding which we found, but also we found that actually these free-tailed bats have been tested positive in serology by some teams, that they were already discussed at the origin of the Sudan outbreak with Ebola Sudan strain. There lived lots of these bats under the roof of the cotton factory where it started. And Botswana has also infected this species of bats with Ebola and show that they can get infected and shed the virus for a certain time. So that's all we have. I'm not saying that this was the origin of the outbreak. I'm just saying it's interesting and pointing into a direction which one should also look at more closely. And all we can say is that there was probably no large outbreak in the wildlife because there is no large wildlife next to the village, and the wildlife which is in the area was not affected. We cannot exclude obviously individual cases and things like that. But then the question would be, how does this body make it into this remote, into this Biliandou village, which is not on a major traffic road to Conakry or something like that. So it's all quite unlikely. So the exposure of the kids was interesting because there is contact possibilities to bats. And looking at all the data published and our own data about the prevalence of these viruses in the wildlife population, especially the potential reservoir population, it must be an extremely rare virus, and the populations of bats, especially if you consider multi-species possibilities at a host, the prevalence, if it's quite low, if it's only 5%, I don't know when we will ever make it to sample enough bats to really find the virus. It's a bit like trying to find measles virus in Berlin. If you sample 1,000 Berlin people, you will not find measles. Still, we know it's there, it's circulating. So perhaps it's a matter of stochasticity and just bad luck to get that epidemic started. Anyway, we are now interested in which small mammals are actually adapted to human settlements, to look a little bit at the possibilities of contact between humans and wildlife, which has followed into the human settlements. There's a more about this stuff you can find in an issue of National Geographic. David Coleman came with us and did a little piece of our work. Now the second great ape killer, and I will also present some data which have not been published yet, but they are hopefully coming on next year, is something which we would consider anthrax. So when I was there in Time National Park in 2001 to 2002, I saw the chimpanzees just dropping dead in front of my eyes. The male was still fit in the morning, beating up his women, having sex, everything a good chimpanzee man has to do in the morning. And then he ate something and poof, he dropped dead in front of my eyes. I did those necropsies and we found that it's something like Bacillus anthracis. Later we found another outbreak in the Jar Reserve in Cameroon. And looking at the bacilli we isolated, it's actually Bacillus cereus, but it has a 2+me, which are characteristics for Bacillus anthracis. And it's on those 2+mes where all the pathogenic properties of anthrax are actually encoded. So we don't have a chromosomal background of anthrax, we have a different chromosomal background, but we have those 2+mes which turn it into a killer. And we have worked a lot on this subject over the last years to really characterize these bacteria really well. And what we obviously also did is, since 2001, is to do all these necropsies on all animals we find there in Thai National Park. So we have done more than 147 full necropsies on animals we found there in the forest. We collect all these bones, and the bone collection even dates later back to 89, because the anthropologists already collected all these bones, and we're collecting blowflies in the area. With that material, obviously, we do molecular analysis, bacterial cultures of the species are positive, and then we do the full genome. So we have more than 200 full genomes now from the area, from this funny, weird bacterium. We're also doing short-term studies. This is a PhD subject of 2 of my PhD students, where we did a snapshot. This is the Thai National Park, the whole blue thing. And this is a research area, so we made them collect every kilometer along a grid system flies. I put up a fly trap through all this area, and then we analyzed those flies for anthrax, and about 5% of these flies are positive for anthrax, and so we culture and sequence the thing and do the SNP analysis. And so we can have a nice map of the distribution. And the other thing is to do a zero study. That's why we're catching the duikers and other animals to see actually exposure. On the large scale, we were interested if this lethal Bacillus cereus is also found in other areas. We had first hints from Cameroon that it killed chimpanzees and gorillas in Jar Reserve. So in collaboration with all the different partners we got more samples. We got blowflies and bones and necropsy samples. And we always perform the same kind of analysis. And this is a paper we just submitted where we can show actually that besides Thai, we find it in Liberia, Jar, I mentioned, and then in Zanga, in CAR, and in Loebo in DRC. And we did the full genome analysis, SNP analysis of those strains, and they're all closely related. They form a new cluster of Bacillus cereus causing indeed anthrax. Here is the anthrax clade, and here is this new Bacillus cereus biova anthracis, how we call it, clade. And we're really interested in getting broader screenings done and perhaps sensitizing people in the different countries for thinking about this pathogen as a possible source of death or disease.
Did you do the sequence of the Parthenon, the anthracis that we get from Senegal, we just published last year? We get 3 genomes. So you include that, yeah.
Yeah, but
so
they are in the
should be very close, huh?
But they're in the anthracis clade.
Yeah.
Okay, so that's anthrax, still a subject where we have many, many questions, and there are 2 Ivorian and 2 German PhD students working only on that thing. So it's really an important issue for us. The third subject I want to touch respiratory disease, and this is in red here because the causality is different from what we discussed before. Our worry when visiting the wild-grade apes and working on their behavior, or we're doing tourism, which we are not doing with others, on wild-grade apes, is obviously that there can be an exchange of microorganisms or disease between humans and wild-grade apes. So we looked at samples which we had from various outbreaks in Thai National Park, and we by now have also samples from other outbreaks in other countries of Africa with a similar setting. And we analyze those samples, and we always find paramyxovirus, pneumovirus, respiratory syncytial virus, which is closely related to human viruses, and Streptococcus pneumoniae, which is actually also a very interesting subject, but I won't talk much about this here, I'm still working on it. And human metapneumovirus, which is very close from the symptoms and even phylogeny to RSV. So we did the phylogeny, and we could really show that the strains we found in the chimpanzees, and we confirmed that now with other findings, are very likely of human origin. So the primatologists are working with, and the conservation people, they said, oh, shall we stop now research and tourism, because we are threatening, we're killing the animals we want to protect and we like. And this is where we as infectiologists have to look a little bit outside our box and and look at the broad picture. And we also had data on the poachers, the density of poachers in Thai National Park. And so you see that here around the research area and around the Giro tourist area, which Bernhard knows as well, there are much less poachers, and the chimpanzee density is also higher in those areas where we were over the tourism happening. And this has been also shown for other sites, so the protective effect of research activities and conservation activities like tourism is obviously much higher than the damage we do to them. And we can do something against this damage. So we now implemented a 5-day quarantine for everybody before seeing the chimpanzees. We are wearing face masks, we're keeping a distance. Everybody has obviously to be healthy and vaccinated. And from that, we got to the point where we developed some kind of employee health program, because the less health problems you have in your people, the less afraid you have to be, and less people drop out of the quarantine. But since people are living somewhere, they're living with their families, where they're going every 2 weeks, we are also starting, we have started to look at the village health in general. And this is obviously not only from the conservation point of view, but also for the people themselves, that can try to enhance the health status in these areas. Now I have a little green point here, because the green are the naturally occurring or probably somehow originating in the habitat microorganisms, and the red ones are introduced by humans. So we also found, by doing all this necropsy, a new tuberculosis isolate in a wild chimpanzee, which is in the whole phylogeny of these TBs. It's really basal and has not been described in humans before, but it's obviously an interesting hint, and other teams are now looking at this strain is also found in people in the area or in other areas. So here again, the chimpanzees pointed out an interesting novel, or not known today, organism to us, which we can now investigate in the human population. If we look at the diseases, the microorganisms called chronic or non-severe diseases, there's an example of SIV chimpanzee, and these are not my data, these are mainly studies done by the Bombay team and Patrick Hahn, and you probably have seen this paper, that SIV-infected chimpanzees tend to die earlier than the negative ones, and they also have a certain pathology associated with that. And also this study was done very similar to what we do through long-term observation and feces collection and urine collection from wild chimpanzees, in this case in Gombe, Tanzania. So there is obviously a disadvantage of being infected, and I was not extremely surprised by that finding, because we all know that chimpanzees are hunting other primates, so there is a risk of SIV transmission from other primates to the chimpanzees. And in the case of SIV chimpanzee, it's even a recombinant virus originating from 2 different monkey species. So it's even an extremely weird construct, and I won't go into detail here, but this weird construct obviously leads to disease in those chimpanzees, not as severe as what we see for HIV in humans. So that's quite interesting. And if we would behave as chimpanzees do, we would all be infected. So it's also interesting why some individuals still stay negative despite high exposure. So I think there's lots to study still, but at least this is what we know until now. The next microorganism I want to mention, just to show you how much we care about virus, bacteria, parasite borers, is malaria. So we found Plasmodium in wild chimpanzees, other teams did that as well. And they are infected in quite a good prevalence. So we looked at chimpanzees from different age groups from Thai National Park, and could show that the younger chimpanzees are more likely to be, I don't know if I should call it infected, but at least they're more likely to find DNA of Plasmodium in their feces. So there is an age effect, as we know, exists as well in humans. Then we, through all these years of data, sample and data collection, we also know which chimpanzee was pregnant at what point. So we have samples from pregnant chimpanzees as well, and we could also show a certain effect of pregnancy on the Plasmodium detection probability in feces. And that's actually also published by now. And we also have first hints by looking at all the data for some individuals being more often positive for Plasmodium than others. So there are probably also inter-individual differences, as we know also exist in humans. This is some people who get malaria all the time and some who have almost no problem. And this is not our studies, this is all experiments done in the US. We know they can develop clinical symptoms when we infect them experimentally. So what we are doing right now, and I don't have data because the study is ongoing, is we're trying to look at the influence of Plasmodium infectious on chimpanzee and gorilla health by combining these different data sets. So we're looking at fatigue by observation, we look at play behavior, feeding, traveling, and so on behaviors. At the same time, we collect urine samples, which we capture from HRE, which is an inflammation marker, linked also to Plasmodium infection in humans. Hemolysis we can do through also testing for hemoglobin with urine samples, so it all has to be non-invasive. And fever, we developed a test where we can get fresh poo, you know exactly when the poo dropped from the chimpanzee on the ground, and you go there, pick it up, and you measure the temperature decline. You can estimate T0 and say if that chimpanzee had fever or not. That works quite well. We published this method also some years ago. And obviously we can detect the microorganism itself, Plasmodium or PCR. We can determine also then by sequencing which type it is. And so we hope with that data set we will get some idea of the potential mild impact, especially on the younger chimpanzees. Now again, respiratory disease, they're not always severe, they can also be mild. And we have no clue why this is the case. We developed a tool where we can find the respiratory viruses in non-invasive samples from chimpanzees, and now it also works for gorilla and bonobo. So we can say this grade 8 is mildly sick, we collect the poo and we test and we can describe the virus behind, but still it doesn't turn into a severe disease. So there are obviously many factors which may be linked, that building up immunity, the influence of secondary infections like Streptococcus pneumonia, but also other factors as described for humans. So we are trying to find out now in a broader study what are the drivers for severe respiratory disease in wild grade apes, and yeah, that's quite a challenging subject as well. And then we have a whole list of viruses, bacteria, parasites, mostly probably original grade A microorganisms, but also to some proportion transmitted probably by humans, as shown for E. coli, for example, shared between humans and gorillas, sharing the same habitat in Uganda. So all these things, we have no clue really how they influence health. And this is a list, a table, which we published in the special issue of CMI, where we just listed all the viruses described so far in wild grade apes, or at least at that point in 2012. And I marked those where we know the clinical importance, and for all the others, we have no firm data on any clinical impact. Some, like foamy-virus, for example, are also not important in humans, we know that, but some can clearly be linked also to disease in humans. And I think we need this long-term observation, long-term data collection to perhaps pinpoint any clinical importance of any of these viruses. And the list for bacteria would be as long as this, and parasites even the same. What I want to show here is that I'm always amazed that these are the killers we know, besides bullets, which is also not healthy. But if we think about microorganisms, these are the microorganisms we know causing disease or severe disease or death in wild grade apes. And if you think about how many different pathogens are killing us as humans, I think that can't be all, and we really have to do more systematic monitoring of wild grade ape populations, because it will also lead to the discovery of interesting microorganisms which may have a high potential to be important for humans. It gets even worse if you look at those diseases with a rather mild impact. There is virtually nothing known. And in humans we know that's a big proportion of the health problems we have, a mild disease. They keep us from going to work, they're not good for the economy, and so on. And for our position in the whole social structure, so So I think there is still a lot to study here as well. And then the third complex is obviously to find out if any of these things here could match. I want to move to a small needs, as I call it, section. So as I just said, I think we need to find out more about microorganisms which matter. And where are actually the respiratory microorganisms which naturally occur in grade 8s? Where are the grade 8 rescue viruses? Don't they exist anymore because the population is so fragmented that the virus died out due to our habitat fragmentation? Or did they never exist? Because we also know that in human evolution, the big diseases came up when the humans were grown together and bigger accumulations of human occurred. So perhaps evolutionary, we are sort of before the human civilization stage here. So I think that's a really interesting thing, and the grade X may mirror a little bit our health problem, health burden we had before we lived in bigger crowds together. So these are some speculations which are fun to talk about, especially over a beer. I think we need many more data to really get firm ideas here. So I'm calling for more necropsies and train people to do that in the same way. So we do it no matter how bad it is to do a necropsy on a chimp, which you know for 25 years or 15 years or whatever. And obviously this has to be done under the highest possible safety measures. We did have cases of Ebola in our chimpanzee group in Time National Park. We haven't seen it since '94. We know about anthrax, we know about the potential other diseases in chimpanzees, like in Sutimengabis, we found monkeypox, for example. So you have to protect yourself and your staff and train them well. But then for looking at the microorganism called rather mild chronic disease, this is nothing you do by going in the forest for 3 months and collecting some piles of food. This is where you need good collaborations with primatologists who know all these individuals, and they all have their name and their history and their friendships. Yeah? So this is Sagu, and this is Sumatra, and this is Shugun, Miriam, Makeba. We know them. And if you look at their faces, you will see they are different. And it's like a family story. And with that cohort study, basically, an animal cohort study, grade 8 cohort study, linked to sample collection during feces from those animals, that's turning into a treasure, which will allow us to study very interesting questions. Non-invasive sampling, as I mentioned, can be done during feces. And the other thing is to talk to primatologists, because they say, oh no, we don't have any disease problem. But if you ask them again, or you give them something to drink, and then you ask them again, then they say, oh, actually, you have the Sutimangabee, which doesn't have a nose. You know? And then, I mean, they're looking at this thing and saying, we don't have a disease problem. And then you should just invest a little bit in a ticket and go there and anesthetize that monkey and take samples, and you find out that this, in the case, for example, it's a very nice treponema infection which just blew away the whole nose of this poor monkey. She's called Phantom. Or this one here, which was also feeling very sick over a few days, very tired. We're still trying to find out what happened here. Then the second bullet point here is a transmission of microorganisms between species and among species. So we can't think about those grade 8s. You know, we used them for years, well, especially the Americans used them for years for animal studies in the US, infecting them with influenza virus, with what other things, just to see what happens because they're good animal models. But at the same time, they exist and they run around the rainforest, and they are like indicators or pathogen samplers for us. They're like perfect volunteers. And they do things which are very similar to what we are doing. So for example, chimpanzees are big bushmeat hunters. And in Thai, they only hunt other primate species, they don't hunt tigers. In other areas, they also hunt antelopes. We don't know why that difference exists. We know that bonobos eat a huge range of different mammal species, and we know that gorillas don't eat meat. So we have a perfect study design, basically, set up by nature. So what we did in the last years is to look at retrovirus transmission from prey to hunter. So we know that the chimpanzees, from our behavior studies, that they eat about 180 grams per day of meat. We know that the mostly hunted prey are these red colobus monkey. 80% of the prey are red colobus monkeys. These monkeys, they're also easy to dart. They're really stupid. They have not learned in evolution that it's a good idea to run away when a predator comes. So they deserve to be eaten. And we have samples from these red colobus monkeys as well, because we guarded some, and we collect also the remains of the meals when the chimps had eaten one of them. Then we find drops of blood and a little piece of blood. So we collect that stuff and we can use it for our analysis. So we found out that 80% of them are infected with SIV, Western red colobus monkey, a species-specific strain as usual. So a chimpanzee eats 45 kilo of SIV-infected meat every year. Or a big male who eats even more meat would, like, at the age of 30, have eaten 675 kilo of SIV-infected meat. So we looked in the chimpanzee samples which we have for SIV red colobus specifically, and for all other SIVs known to occur in the area and known to exist. We did that with PCR methods, and we did that serologically with Martin Peters from Montpellier, who you probably all know. And we could show that, in fact, there is no transmission happening, despite this high exposure. And talking to people who are much more into the virology than I am, also on the molecular level, it's not possible for the virus to infect the chimpanzees. There's something with Appleback and Neff and so on happening. Why? That actually doesn't happen. But I think it's nice to look at real-world data, because we are not sure if we understood all mechanisms of transmission, and to confirm these theories we have for the molecular findings. I don't want to go into detail of all the different things we did about the retroviruses, but basically what we did over the last 14 years, and it's summarized by a PhD student of mine in the review, is to look at different monkey species, which viruses, retroviruses and others, it's just retroviruses here, they carry, which of those are transmitted then, so we do specific tests in the hunter for those viruses, and also in the humans, in the hunters. And so we could show that STLV1, so simian T-cell leukemia virus, is transmitted from the prey to the hunter. Simian Fabien virus is rarely, but it is transmitted from these red colobus monkeys, for example, to the chimpanzees. And that the chimpanzees have their own simian T-cell leukemia virus and their own simian Fabien viruses in addition. And that, again, those are transmitted partly to the human population. One interesting thing I want to point out here is that the Sutimangabes, which are infected with SIV Sutimangabes, obviously, that we find that virus in the human population turning into HIV2 in the human population. So we've described a new type of SIV, HIV2 in the human population. But the chimpanzees don't get infected with SIV Sutimangabes, even though we can infect them with HIV2 in the lung. And this is when you need your primatology colleagues again, and the answer is really simple. The chimpanzees don't eat the Sutimangabes. They're living in big rooms, they have big teeth, they're too dangerous. So it's the behavior which avoids transmission. It's not any genetic factor or something like that. However, I'm a bit worried, or also a bit curious to be honest, being a somehow scientist, is that the red colobus monkeys are also hunted a lot by the poachers, and the populations are declining. So if the chimps don't have the easy prey anymore, they may move on to other monkey species. And if they move on to Sutimangabes, that could result in the end in HIV2, SIV Sutimangabes infections actually in the wild. So there may be shifts due to human influence. Then I want to mention also transmission within the species. So what are the transmission dynamics of certain viruses, other microorganisms within the species? So we looked at simian viruses, which we find in high prevalence. You can find it easily in feces sample, which is very convenient if you want to work non-invasively, and they're known to be normally species-specific. So again, we took parts of feces from all these chimpanzees, and we selected mother of offspring sets of samples. So we have from Sumatra, Shogun and Cetu, for example, and from all the other families. We took the parts of feces, several obviously over the different time periods, and looked at the fomivirus diversity in those individuals. And we could show that the family, like Coco, is in the mother, Caramel, Celine are the offspring. That here, Coco and Celine are sharing the same strain, but Caramel got infected with another strain. And so we could do that for all the different families we have. And with the statistics summarizing all that, we could show that the chimpanzees are more likely to get the fomivirus from their mother in an early age. And then if they get strains later, they get them from any other member of the community, no matter if it's a father or someone else. But also that once they have the strain from the mother or not, that with life they accumulate different fomivirus strains, and this is because they enter in the social hierarchy, there are more fightings, more blood-blood contact possibilities, so they are accumulating the diversity of fomiviruses. Last point are innovative methods. So I know that's a little bit crazy, but I had the idea that we should be more efficient in finding carcasses. It's extremely rare to find a dead animal in the forest, and it's a lot of work to find them. So I was thinking, who could help us? And I was thinking about sniffing dogs or rats and things like that, but that's extremely challenging. I still want to follow that path. So the blowflies, whenever you find a dead mouse or anything, the blowflies are always there first. So I asked my deputy, Sebastian Spencer, an excellent French scientist working with me in Berlin, which is very good for me, if we cannot find out what kind of mammals the blowflies have been feeding on. And we could actually show that these carrier feeding flies are great samples of biodiversity. So just by looking at 100 flies, you can find out which primates live in Thai National Park. And so, okay, the president of my institute said, oh, it's nice, we have a carbon molecular ecology and so on, but what does it have to do with infectious disease, what we are supposed to work on? So we obviously moved on to the infectious disease side as well, and as I showed before, we're using the system to look for anthrax already, and that works really nice. But we also looked at other viruses. So for example, we took 500 randomly caught flies, selected 143, which are mammal positive, and we looked for adenoviruses in those flies. And it's not a great yield if you think about it, but also to catch 500 flies is actually a matter of a week work in the forest. And if you do the phylogeny of those adenoviruses we found in the flies, they're here with the primate must adenovirus, so they really got it either from carcass or from feces of the primates existing in the area. And we could add a few strains here in the phylogeny of primate adenoviruses by using the flies as samplers for biological material. And we're still following up this path a little bit, and I think it really depends on many factors if you find something or not. It depends obviously on the stability of the pathogen, if it's DNA, RNA, when the fly has fed, how much it has fed, and so on. So I'm not saying it's a wonderful solution to all the problems we have with sampling wildlife, but it's an interesting approach. And then the last point, I think that's quite clear from what I have shown you in the last slides. I'm a really big fan of cross-disciplinary, interdisciplinary work. To work with primatologists, geneticists, physiologists is really what's helping us to move on. And I forgot the anthropologists, which are also extremely useful now to think more about the human side. So, but that's for you, as I heard about the structure of this unit, it's already there. You're doing a lot of that. Okay, I want to thank my research group because I'm not doing this all alone, especially Sebastian, Calvin Spencer here, and then there's a number of postdocs. This is just the funding we're using, so our RPI is funding a little bit of what we're doing, and the rest is like German Research Council, Ministry of Research Education and Science. We have a very strong capacity building aspect also on all our projects, so there are always students from Ivory Coast, from Gabon, and from DRC involved in the analysis, and we are moving more and more to doing most of the screening, at least, PCR serology in the country. So we have really strong partners in Ivory Coast, where we're mostly working in the lab, but also INRB in Kinshasa is a very strong lab, as probably some of you know. So we're trying really to do this together. Other partners I want to mention, well, the African partners I just mentioned already. Then we have a few partners in the US. In France, we are mainly working with teachers for the retrovirus question. And in Germany, very important partners is the Max Planck Institute for Evolutionary Anthropology, because they give me access, and well, I'm co-director for the Grade A projects for responsible for health. but also the WWF is more and more interested in working with us. And then the other partners are individual projects where we're interested in filoviruses, so we work with University of Marburg at Becker, or hantaviruses at Krüger, parasites with Block Institute, retroviruses, and so on. So for each subject where we are not specialists, some things we do ourselves, but if we're not specialists, we always ask help from outside and do it together in bigger collaborative studies. That's why on most of our papers you will see many co-authors. And I don't think that's a crime. All right. So this is it from my side. I want to thank you for your attention, and I hope there are a few questions.
I think it's a very clever idea to go and take some flies, biting flies. It's a bit done on ticks and mosquitoes, but here to collect flies in the wild to look for blood in monkeys. It's a great thing. We have worked a little bit on Apes, and the bugs that we find so far, we find Rickettsia quite commonly. We suspect that we get several line of evidence that Anopheles are transmitting Rickettsia, and probably if you get malaria, when we link malaria and Rickettsia everywhere in Africa, so I suspect that they get Rickettsia as well. So if you look
it's really Rickettsia, the same strain, or
yeah, Felis, yeah.
Okay, and then the samples are from areas where there is contact possibility, transmission possibility, or are the grade A samples from remote populations?
The ones that we get came from Cameroon. So the ones that we tested first.
So still, you have to see where they're sampled, because I think there's obviously a flux of pathogens back and forth at the periphery of parks as well. And then some of the geography is really
I'm pretty sure that anywhere where you get Plasmodium, you get Rickettsia because it's the same vector.
They have not found the vector yet for all these grade A Plasmodium.
Oh yeah, but
it's like
the other thing that we find recently in Cameroon as well was Leishmania. And one of the basins is we get. It was really interesting. I'm really interested. We were discussing with Michel what the legend is and what the observation is different. So when people say they are not sick, when we start to work with Paddy on the eukaryotes that we can find in gorilla stool, we find plenty of insects. And people say, well, let's say don't eat insects, they use termites and so on. But this year we were together to see gorilla and discuss with the people, say, oh yes, I love this. For example, the tsetse flies, they eat them exactly. So plenty of insects, but in fact, it was not in the book. But when we find that by molecular testing, say, well, they eat a minimum of 10 different kind of insects. So in fact, probably one of the ways they may have been able to eat some of the Leishmania. And the third one that we found is Tropharema wordplay, that we find also in the stool. But there is something, I get I'm not working directly on Ebola. We're helping people to do diagnostic on place, but I get a theory to understand how it comes from what you describe to the town. And the biggest change that I've seen in Africa since 20 years of all, the most important change in the ecosystem is cell phone. And I think that there is millions of cell phones, and all the people go together to a source of electricity where they can exchange, the virus of the phone. And the virus is surviving very well on the phone. So I think that all it's come from the forest to the town is gone with the cell phone. I think this is probably why the epidemiology changed so abruptly. You may have something ecologically different, and one the biggest difference that I've seen in Africa since years is this, and this year is motorcycles. This is the second big thing that I've seen. It was cell phones in 10 years, and since 2 years, a Chinese motorcycle, which is changing everything. People are moving everywhere.
But when I heard about the outbreak starting in Guinea, I said, well, that's a mega disaster. And I know that Pierre Formonti, who I know because he worked in Type 4 maybe at Liverpool, he also was really worried already. because it's not an outbreak in the middle of DRC. Like last year there was an outbreak, and it will affect 3 villages, which are a neighborhood. That's bad, but that's all. It will not spread further because it's isolated. But this corner, like Guinea, Liberia, Sierra Leone, with a motorcycle, within 2 hours you're everywhere, you know. And so that's really.
It's motorcycle plus cell phone.
Yeah.
Cell phone is a very. I'm sure you've seen in many villages there is no electricity, so they provide electricity for money, 2 hours. Yeah. So you get all the villagers with their cell phone that put that for 2 hours, and then they touch them. Usually they don't leave themselves because they live in concession. But with these, this is a point, a hot point of meeting where they can exchange viruses. So it's going to change the ecosystem altogether.
Yeah, the epidemiology is, probably. And then there's one point in the village where there's reception, so everybody stands and tries to get reception.
So this is why you need to go a place and observe things to understand. You cannot do that in the, like they do sometimes in the US. They modelize things in Washington, so that doesn't work.
I agree.
The taxonomic classification within the genus, the Bacillus genus, is not very reliable. So, and your Bacillus cereus and Tracis strains appear to form a very strong cluster.
Yeah.
So, might they represent representatives of the new species, or are you confident that they are Bacillus cereus strains?
The phylogeny is just on the chromosome. It's not including the plasmids. So they're really closely related. The plasmids are a little bit different from the others. They're more closely, but we don't have enough strains to do solid phylogeny at the moment. That's the issue. Yeah, we have lots of friends from Ivory Coast, but they're all from the same area, so that doesn't help much. Another question?
Fadi, tu vas parler des gorilles. You work on gorilla.
I know, we met in Berlin once already.
I have only one question. If you look, most studies are focused on apes in Africa, but no studies from Asia. Did you have any question, any reason for why you have not any data from Asia or?
Right.
Yeah.
It's really a very interesting question. So, but if you look in Asia, so the orangutan, they are solitary. So there are very few wild orangutan habituated because it's extremely boring. Just go to the zoo and you will see they're just sitting there. So the wild orangutan are not as well studied, and there are extremely few left as well. So where you find lots of orangutan are the sanctuaries, and they're really full because the habitat loss and they're confiscated. And so there are huge health issues in the sanctuaries, and there are some studies done in that respect. There are some teams working on that. But there's always a question of directionality of the pathogen you find. Is it acquired from humans or is it really their own? So that's a bit boring. But I agree it would be nice to have the orangutan because they're a wonderful outgroup, like the Asian great ape and then like microbiome or whatever. I have 20 samples from wild orangutan. So can talk about that. But it's extremely hard to get this stuff. Other questions?
Is there any evidence for the role of transmission of Ebola by arthropods?
There's no good evidence.
Maybe mosquitoes, ticks, or...
People were afraid of that, but I'm not aware of any good description.
Did somebody look in the ectoparasite into that? It's anybody look in the ectoparasite of the bat?
No, but everybody talks about it, but I don't know if anybody does it. So the bats are super interesting ectoparasites, you know this.
Yeah, it's easy to look at.
Yeah, we have a few of these, and can have a quick check, but I think also here the question of sampling depth is a really critical one. So I think I'm not a big fan of animal experimental studies, but I think we should go to the lab, to the BSL-4 lab, and in fact candidate species of fruit and insectivorous bats. I know that the Americans are doing it for fruit bats, and we have plans for the insectivorous bats. You know, a new BSL-4 lab to really look at transmission between bats and the biology of the virus in the bat goes.
That would be a good idea to parasitize them with ectoparasite and
Yeah.
see what the role should be. There is no reason why it should not.
We're always collecting them, and I can tell my other colleagues to also do so.
But from Ebola infected bats?
Well, there are no Ebola-infected bats in any collection. Eric Lavoie has those few samples, and he doesn't have the parasites, I'm sure. He doesn't even find the original sample. So that's the thing, you know. But perhaps for the Marburg, we should use Marburg first. But I think Towner has, I'm not sure if he has looked at that. I have to look that up. But he would be the right person for a very similar virus to have a look at.
Or lice, look at lice.
Yeah. They have these bat flies, you probably, these wingless things. And if you look at the size of the bat and the fly, it's like if we had a hamster, like parasiting one of us running around. So they're really big predators. So, I'll check the literature for Marburg.
Okay. Good, thank you again.
Thank you.